Hi, this is Siranush. The reason for creating the modification for the cow milk is important because lactose intolerance has become a highly prevalent problem in the society. There are some people who are lactose intolerant. They are unable to digest milk or lactose because of the absence of lactase in their digestive system. Often lactase is produced during infancy so as to digest the mother's milk but ends after weaning begins. After this stage, humans become lactose intolerant to various degrees. Lactose is a sugar that some people cannot digest. More people cannot consume milk or other dairy products with lactose without experiencing health problems. Therefore, making the modification is beneficial as this large number of people will be able to consume cow's milk. The negative impacts have not been outlined to show that the process results in harm to the animals or cause any suffering into the cows. Through the modification, most people will be able to easily consume cow milk without developing stomach problems, as it is in the case of lactose intolerant people. Furthermore, the milk that is created following gene modification is likely to be healthy for human consumption. So, by consuming this particular milk, humans will not be having stomach problems or develop any other health conditions. This new milk can also be consumed by those who are lactose tolerant. While ordinary cows produce milk with lactose, the milk that is produced by the new cows does not have lactose. Hence, it can be easily digested by lactose intolerant people. This occurs because the gene that is introduced into the cow produces enzymes that digest lactose sugar in cow milk to other types of sugars that are easily digestible. These newly formed sugars can ease digestive disorders among the lactose intolerant people.
By the way, this is Jacqueline. Lactose intolerance, also known as lactose malabsorption, is when a person lacks or does not have enough lactase in their stomach and small intestinal lining in order to digest lactose. Lactose is a type of sugar that is prevalent in many dairy products such as milk, cheese, butter, and ice cream. These dairy products are common and in prevalent use in daily life. Lactose intolerance can cause symptoms of bloating, diarrhea, and gasiness after consuming foods or drinks that contain lactose. Symptoms of lactose intolerance generally do not occur until there is less than 50% of lactase activity, and this is important because lactase is responsible for breaking down those lactose. With the lack of lactase activity, these symptoms occur due to abnormal processing and absorbing of lactose from milk, which is instead passed into the colon where it's fermented. And while in the colon, the bacteria that reside there interact with the undigested lactose to cause these symptoms. Even though the condition is seemingly harmless, the symptoms are usually uncomfortable and can last from 30 minutes to 2 hours.
Hi, this is Carter, and these are the steps to create the bovine zygote with lactase. The first step is to isolate the bovine DNA from bovine tissue such as steak. The plasmid to be used for transfection of the bovine zygote is PEQ176, which has and expresses the lactase gene, also known as lactase, and its CMV, which stands for cytomegalovirus promoter. The plasmid also contains the HIN3 restriction enzyme and BSRG1 restriction enzyme sites. We will be using the BSRG1 cutting site and the HIN3 cutting site. The first 6 nucleotides are the restriction enzyme recognition site, and the remaining primers were designed by the Mozani team, who we would contact for their permission to use their primers since they are doing independent research on this topic currently.
Hi, this is Christopher. Using this restriction enzyme, we will cut out the CMV promoter, giving the plasmid sticky ends that will later be used to attach to a beta-casein promoter. To isolate the beta-casein promoter, we will cut it out of the bovine DNA with restriction enzyme BSRGI and HIN3. Then PCR the promoter, leaving it with sticky ends to attach to our plasmid that had the CMV promoter cut out. So to complete the desired plasmid map, we will combine the cat plasmid and isolate bovine casein promoter with DNA ligase in a test tube. Then we will transform E. coli via heat-soak method to grow our desired plasmid on a agar plate. Finally, we will extract the zyoplasmin from the E. coli and transfer the bovine zygote with the plasmid. The zygote will be allowed to grow, and then the milk will be analyzed for presence of lactase and hopefully absence of lactose. This plasmid will be considered under the proof of principle. I don't know. And what is it called?
My name is Catherine, and I'm going to talk about the ethical concerns of our project. With the advancements in genetically modified organisms, there are often ethical concerns that are raised that pose the question if the benefits really outweigh the risk. For our proposal of modifying bovine to produce lactase enzyme in the udder, there are possible ethical concerns that may arise in how this modification will potentially positively and/or negatively affect the bovine, humans, and the environment at its current state. The first of these considerations is the ethical concern for the safety and well-being of the bovine that the modification will be performed on, and that of the bovine offspring. This procedure will not be invasive nor harmful to the bovine that the modification will be performed on. A possible concern post-modification is gene resistance to certain antibiotics. This is very necessary in the introduction of the plasmid to the bovine, but it could have negative consequences by causing risk of severe infection. We intend to resolve the resistance to antibiotics if and when it poses a problem. Another ethical consideration is if the benefits to humans will outweigh the possible risk it poses. With this approach, we will successfully be eliminating the presence of lactose in the final milk product without having to do further modifications and processing on the milk product from the bovine. As expressed earlier, there is a significant population with intolerance to lactose. It is estimated that 65% of human adults suffer from lactose intolerance. There are no expected negative consequences of this procedure on human health and well-being. Therefore, we believe that it poses significant benefit. The impact that this procedure may present to the environment is another ethical consideration. With a single cow releasing between 70 and 120 kilograms of methane per year, it poses a concern for how this modification will improve and/or worsen the bovine contribution to greenhouse gases. Studies have shown that undigested lactose remains in the intestine and is fermented by colonic bacteria, producing hydrogen gas, carbon dioxide, and methane in some mammals. Although further tests would need to be performed to prove this theory, we believe that the modification has the potential to improve bovine contribution to greenhouse gases.
As stated, the goal of recombinant bovine is to create lactose-free milk for the lactose-intolerant population. By inserting the lactase gene downstream of the beta-casein promoter in a cow's genome, whenever casein is produced, lactase will also be produced. Casein is a protein responsible for making the cow milk white, and it's only produced in the cow's udder, which is the source of milk. We chose beta-casein in particular because it is the casein protein that is present in the highest percentage, which is about 30% in cow milk. By using beta-casein, we ensure that the lactase gene will be expressed at the highest percentage possible, which ensures an adequate amount of lactase production to react with the lactose. Due to our recombinant DNA, lactase enzymes in milk, which contain lactose, will be produced simultaneously. Therefore, lactase will readily break down the lactose, creating lactose-free milk within the cow's udder.
Hello, my name is Andrew, and I will be discussing the promoter and protein sequences for beta-casein. The promoter sequence is about 1900 base pairs long and was found June 24, 2018. In the document itself, the promoter sequence has their stop and start codons highlighted, while the protein sequence was found the same day and is made up of 336 amino acids.